So we are studying the mm, tissue engineering process. We are studying the tissue engineering process. What happened? That we have taken a stem cell from a from human body by biopsy, and then isolated the stem cell and cultivate it, and then uh, 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 mm, pol- proliferate it. Proliferate means from one cell to two cell, two cell to three cell. That is called proliferation of cell. And this stem cell is seeded in a scaffold uh, where tissue developed. Here tissue is developed. We have given the or they have given the appropriate environment to develop the tissue. That is called mechanical stimulus, and then the whole scaffold is implanted to the human's body. This is the tissue engineering. What are the tissue engineering? If I see here, process of tissue engineering, start building material, that is extracellular matrix biodegradable polymer, which we call it a scaffold, which we call a scaffold, a building material, which is an extracellular matrix. It is not not from the cell. It is extracellular matrix, which is biodegradable. That means which will degrade after a definite period of time. That is called biodegradable. Now, shape it as needed. Shape it as needed. Shape the extracellular matrix. Shape the scaffold as you need it, as your desire. You shape it and then seed it with living cell. And seed it with living cell means place place some living cells to the scaffold so that it can differentiate, or it can duplicate, triplicate, multiplicate, and but it with growth factors. Growth factors means, what is growth factors? Growth factors means it's a special environment. A cell requires its special environment to grow. So let them dissolve. Let the scaffold dissolve in that special environment. Now, what will happen? Cell will multiply and fill up the scaffold and grow into three dimensional tissue. Then, when you have placed the appropriate growth factor, appropriate environment to the cell, then the cell will multiply, and it will uh, uh, mm, uh, fill the scaffold, and and the desired shape will come. Then you can implant the whole scaffold with all the tissues in the body. You have to place all the scaffold with all the tissues in the body in the appropriate place of the body. Then cell recreates their intended tissue functions. Then what will happen? Then the cells or tissues will function with the body. Function with the body. do whatever what body does. They will function with the body, and the blood vessels attach themselves to the new tissue, and our blood vessels will attach themselves, will attach our blood vessel will attach with the new tissue. Blood vessel will go through the new tissue to give them nutrition. And then the scaffold dissolves, the scaffold degrades, it will vanish, the scaffold degrade, and the newly grown tissue eventually blends in the in with surroundings. Then the newly born tissue will mm, mm, will will do with it, do work or do whatever its work with the surroundings. Now, this is the whole process of tissue engineering. So, this requires a variety of disciplinary work. Say, for example, they requires this tissue engineering requires genetic engineers. This tissue engineering requires the doctors, medical practitioners. This tissue engineering requires the material scientist and, as a whole, a chemist. Why chemist is required? Chemist is required to prepare the scaffold, to prepare the appropriate scaffold. There are various types of scaffold depending on what type of organ or what type of tissue you want to make, you want to engineer, what type of tissue you want to engineer. So you will require various type of scaffold, and this scaffold is made by a material scientist and a chemist, rather a chemist. And what genetic engineers do? Genetic engineers multiply the cells, and above all, they control the signal. That means they control the gene. Why? How they control the gene? Say, for example, when it start, wh- what type of tissue it will grow. A stem cell will, a stem cell can grow various type of tissue. Every type of tissue can a stem cell can grow. Say, for example, a stem cell can grow your bone tissue. A stem cell can grow your skin tissue. A stem cell can grow your muscle tissue. So, which type of tissue a stem cell will grow, it will directed by the gene. And this direction, this signal is controlled by a genetic engineer. And the medical practitioner, of course, a doctor or a biologist, he, he will place the scaffold in the proper place, and he will see that whether it is working or not. Now, our our interest is on scaffold. So we can talk 1 or 2 minutes with the scaffold again. Say what scaffold does. You see the isolation of cells from human body. This is again tissue engineering. The isolation of cell from human body. That means these cells are stem cells, not all qualities of cell. These cells are only stem cells, and the expansion of cells in the appropriate medium, and then you put them in the scaffold, And after putting the scaffold, you can transplant it to the body by surgery, or you can transplant them by direct injection into the body. This is again the tissue engineering. Now, what are scaffolds? You see, this is on scaffold. This is a very close look. This is 1 millimeter. This is 1 millimeter of thickness. This is the scaffold. So what is scaffold? Scaffolds are three-dimensional materials constructed in order to provide structure to a developing tissue. A tissue will gain which structure? It will determine the scaffold and and to allow the cells to adhere, proliferate, differentiate, and most importantly, secrete extracellular matrix. You know now, adhere means the adhere means proliferate means multiplication of cell. This is called proliferate. Differentiate means to multiply the cell into different types of cell. Different types of cell means a bone cell, a skin cell, a tissue cell, different types of cell. This is called differentiation. So most importantly, secret extracellular matrix. Extracellular matrix means the uh, uh, the uh, the matrix of the matrix uh, beyond your cell. That is called extracellular matrix. Many different materials have been investigated in order to construct a scaffold. How you can construct a scaffold? Which material you can construct a scaffold? So there are some polymers. There are polymers. There are some ceramics as well as natural polymers are there. There by which you can prepare scaffolds. Now, what are the requirements for scaffold construction? Scaffolds should provide void volume for vascularization, new tissue formation, and remodeling. Scaffold should provide void. That means a scaffold should be porous so that so that blood vessels can go, new tissue can form within the pore, and all the tissues comes close together. So scaffolds should provide this provide for provide space for this. Should have high porosity and have suitable pore sizes to replicate, to multiply the cells. It should have suitable pore sizes to multiply the cell, and the pores should be interconnected. The pores must be interconnected. The biomaterials should also be degradable upon implantation. After implantation, after a certain time, the scaffold must degrade. It cannot be there. It should be degrade automatically. So, or a rate matching the new matrix production by the developing tissue. When the tissue is developed fully, the scaffold must degrade. The biomaterial used to fabricate the scaffold need to be compatible with the cellular components. The biomaterials must be compatible with our body. The biomaterial you are using as scaffold must be compatible with our body, and it it also must compatible to the cells uh, which are growing inside the scaffold. Scaffolds provide mechanical and shape stability to the tissue defect. So scaffold must provide the mechanical stability. It should be sufficiently strong to hold the tissues. So these are the uh, these are the scaffold construction requirements. And now, what are the materials that are used in the scaffolding? There are two types of materials, so natural biomaterials and synthetic biomaterials. In natural biomaterials, you have protein origin biomaterials and sugar origin biomaterials. So in the protein origin biomaterials, you can use silk, collagen, fibrin, gelatin as scaffold. Uh, and saccharide containing biomaterials, hyaluron, alginate, agarose, chitosan, they are saccharide containing biomaterials. And in the synthetic biomaterials, you must have polymeric biomaterials, polyethylene glycol, polyglycolide, polylactic glycolic acid, polydiol lactic acid, polycorpolactone. These are the polymers, and these are the ceramics, alumina, zirconia, sintered, hydroxyapatite, tricalcium phosphate, hydroxyapatite, bioactive glass, calcium phosphate. These are the different types of materials by which you can prepare a scaffold. These are some very small introduction of scaffold. Uh, we don't need much. Now, again to the biomaterial. So, biomaterials can be of polymer, biomaterials can be of metal, biomaterials can be of semiconductor materials, biomaterials can be of ceramics, and this it is all I have uh, told you earlier. Now, host reactions to biomaterials. That means how our body reacts with the biomaterials, or what type of reactions do our body with the biomaterials. These are called thrombosis, hemolysis, inflammation, infection, and sterilization, carcinogenesis. This type of infection, this type of your reaction is shown by our body to the biomaterials. Now, for that, we have we must have a material to be biocompatible, so that this type of reaction cannot occur. So again, what is biocompatibility? Which I have told you that biocompatibility is the ability of material to elicit an appropriate biological response. Appropriate biological response on a given application on the body, so that they cannot, a body cannot do adverse effect after you're placing the biomaterial in our body. Now, how biocompatibility is tested? How generally biocompatibility is tested? Two types of testing is there. One is called in vitro and another one is called in vivo. In vitro testing and in vivo testing. In vitro testing means testing in the laboratory. Say, for example, you have made a bone and you have intended to place it in human's body. So what you do first? You will do first place the bone in the exact environment of the body in the laboratory. Say, for example, in the laboratory you have taken a bath where exact pH of the body is there, and you have kept the bone is there. You will see whether there is a change of the bone or not. You have kept the exact your blood composition there. You'll see whether there is a change or not. This is called in vitro. You have attached the bone with a skeleton and see whether it is working properly or not. These are the in vitro. And after testing there, and you have to remind it, you have to test the tensile strength, you have to test the compressive strength, you have to test the fatigue, you have to test the fracture strength of this bone, of this artificial bone you have made. These all are in vitro testing. Now, what will be in vivo? Then if if you see that it is passing all the tests in vitro, then you will place it into a animal body. Say, for example, for a in a big animal's body, say, for example, in a dog or in a pig. So this is now in vivo, and you will see what is the reaction. And after seeing the reaction, you will place it then in a man's body, in human's body for trial. These are called clinical trials, and these are in vivo. These are in vivo. So the level 1 series of preclinical tests in animals and in vitro studies. These studies can be used as a screening phase, but should be followed with more detailed investigation. In uses in test environments at the sites where materials to be used. So you have to place the bone in a animal's body to see whether it is working properly or not. If the bone works properly in the animal's body, then you make the trial to the human. And after sufficient trial in the human, then you can place it to the patient's body. So there is a series of experiments, and it will take 5 to 10 years to bring a biomaterial, to place a biomaterial to the patient body from the date of preparation. So this is the test of test of biocompatibility. Now, during this testing, you have to see many things, such as you have to see the cytotoxicity test. That means if it is cancerous or not. You have to see the sensitization of it, and irritation, toxicity, chronic toxicity, implantation, hemocompatibility, whether our blood is compatible with it or not. everything, everything you have to see after when you are testing this in vivo. When you are testing this in the trial, in the body of human, when you are testing this, everything you have to see. This is called biocompatibility. Now, there are some definitions. What is called biodegradation? Biodegradation means biodegradation means the material will degrade, but it will stay in your body. That is called biodegradation. The material will degrade, but it will stay in your body. That is biodegradation. Bioresorption. Bioresorption means the material will degrade and it will eliminate from your body. It will not be in your body, it will eliminate in your body. As for example, polylactic acid. They are, this is called bioresorbable. So biodegradation and bioresorption, there is a difference. What is biodegradation? It will degrade, but after degradation, it will stay in your body. It can migrate from the place where it was. It can migrate from the place where it was, but it will stay in the body. But bioresorption, the entirely bark degraded in vivo, it is eliminated from the body into low molecular weight molecules. Now, what is bioerosion? Bioerosion means material that first degraded on the surface. The material will degrade from the surface and resorb. Resorb means it will go out from the body. It is erosion. And bioabsorption, what is bioabsorption? Material could dissolve in the body without modification of molecular weight, and a process which in contrast with bioresorption. That means bioresorption is that it will degrade into low molecular weight molecule, but bioabsorption means it will be absorbed to the body without modification of the molecular weight as it is. That is called bioabsorption. So resorption, how resorption works, how a material degrade or resolve by water absorption, by reduction to the material properties, by reduction of the molar mass, by complete loss of weight. By these qualities, a material can degrade or can resolve. Now, how you see? You see, mainly by hydrolysis. Mainly by hydrolysis. Say this is a compound. Now, in presence of water or in acidic medium or basic medium, the bond breaks. The bond breaks by this and this. So it will get a low molecular weight compound, and ultimately it will dissolve or degrade. It will not like this compound. It will grow to this acid form and it will degrade or resolve. So there are many substances. There are many substances which can be hydrolyzed. Actually, many substances they are hydrolyzed due to when they degrade or when they resolve. So degradation and resorption mainly occurs by hydrolysis. Now, next next thing, this is apart from these things, this is called biotextile. Which are the biotextiles? Biotextiles are fibrous textile structures, that means stitches, created from synthetic or natural materials that are used in vivo or ex vivo or in vitro biological environment as a medical device for the prevention, treatment, or diagnosis of an injury or disease. These textile materials, what they will do, they will prevent a disease, they will treat a disease, they can diagnose a disease, and they will help to heal the injury of the disease. What are they? What are they? Say, for example, in the surgery, you will get the sutures, repair mesh. Suture means your stitches, thread stitches, and you have different type of mesh. Mesh means her where a small pores are there. This is called a flat material where a small pores are there. They are called mesh, and then sutures. Now we'll see another type of thing. In cardiovascular application, there are angioplasty rings, blood filters. There are different your cardiovascular instruments or cardiovascular devices that are formed from the your stitches. Dental ribbons, neural guides, bone grafts, bone cements, they all are your, and you see skin and wound dressing. You have dressing, you have dressing. They are also uh, your textile material. So you require different type of textile material in medical sciences. For example, to to make the infected tissue, to make the infected tissue together, you require the sutures, you require the stitches. They are textile material. You require the skin and wound dressing. Everybody of you do dressing. If you cut your finger, you do dressing. This is a biomaterial. These are called biotextile materials. And another type of thing is known as biosensor. This is also a third generation biomaterial. This is a sensor. Biosensor is an analytical device containing an immobilized biological material, that means enzyme, antibody, nucleic acid, hormone, etc., which can specifically interact with an analyte and produce physical and chemical or electrical signal that can be measured. What you do if you want to if you want to measure the glucose concentration? Many of you have a glucose meter in your home. What you do? You take some blood from your finger and keep it in a strip, and then place that strip in a meter, and you will see that blood sugar is 11 in the display. This is called biosensor. So what biosensor does, you are you are interacting the analyte. What do you mean by analyte? Analyte is your blood. You are interacting the analyte with the strip. This is not a paper strip only. It has some enzymes, or it has some antibody, or it has some nucleic acid, or it has some hormone, or it has some mixture where in your strip. So when you place blood in your strip, in the strip, then it will, the analyte will react with the enzyme or hormone, whatever it is in the strip, and it will be detected. It will be detected by your uh, meter, that glucose measuring meter. That is called biosensor, and it will be detected by glucose measuring meter, and it will show the result. That is called biosensor. So what is an analyte? Analyte is a compound that is glucose, urea, drug, pesticide, whose concentration has to be measured. Say you want to measure the urea in the urine, then you require that type of meters. So this is the biosensor, which can which can sense the biological element. Biological element means reaction of analyte with the immobilized biological material. What do you mean by immobilized biological material? They are enzymes, they are antibody, they are nucleic acids, they are hormones. It is placed in the strip, and your blood interacts with that strip, and when you place that in the meter, then sensor sensed it and gives the result of your analyte. This is called biosensor. So what are the components of a biosensor? Uh, it has a sensor, a transducer, an amplifier, a processor, and a display unit. A sensor and a transducer, amplifier, processor, and a display unit. You see the analyte which reacts with this receptor, and then here this is transducer sensed it, and then it is the your what do you call monitor. This monitor will show you the result. So how it works, the analytes, these are the analytes. Say for example, these are the bloods, and this is your strip. This is your strip, and the bloods comes to the strip, and you see here that the blood reacts with this strip, that is called bioreceptor, and the transducer actually detects this. If you want the pH of the blood, then it will detect in one type. The transducer is one type. If you want to detect the sugar of the blood, then the transducer is one type. So this transducer is the main thing, and this gives a electric signal. This signal is detected uh, by the sensor. Application of biosensors. There are many applications of biosensors, such as this: food analysis, drug development, crime detection, and many applications are there. So these are almost all about your biomaterials. We shall see more in the nanobiomaterials. There are another type of biomaterials which is known as nanobiomaterials. This nanobiomaterials also in your it is in your syllabus. So we shall see more in of biomaterial in the nanobiomaterial. So this is all about biomaterial.